Hello, Bartonella buddies. My name is Jake, and I am pleased to be with Dr. Amanda Elam. And she is the CEO and co-founder of Galaxy Diagnostics, which is a medical diagnostics company in North Carolina that offers molecular and serology test options for flea and tick-borne diseases implicated in chronic disease. The first question I wanted to ask you was how, when, and why did you get started at Galaxy? And I also wanted to know how you determined you wanted to use the name Galaxy.
So I came into the picture after Ed Breitschwert and Ricardo Maggi at North Carolina State University had already formed the company and had already chosen the name. But the story behind that, according to Ed, is they were sort of sitting out on the porch one evening, drinking beer and looking, or wine in Ed's case, and looking up at the sky and trying to think, what a good name for the company would be. And they had this, our central product at that point was the Bartonella alpha proteogrowth growth medium, so BAPGM. Mm-hmm. And it grows a lot, not just Bartonella. And so they were just thinking galaxy of microbes. It could grow a lot of microbes. So that's where Galaxy came from. So I came into the picture about a year after they had formed the company, and they had the sample enrichment method. And they were hoping that it would be picked up by, another lab, like a big lab. And they basically shopped it. And the answer was very promising research, but the market's not really big. So I was introduced to them. I was actually working as a business faculty. They asked for some help. They said, can you help us figure out, how we get this up and out there? And then they told me the whole story around the veterinary occupational risk, right? And that's the big story around this is exposure to fleas and ticks and animals who may be infected. Animal workers are the number one risk group for actually becoming infected. I took a look at it, did a deep dive in the literature, determined that this is probably an infection that's much more widespread than anyone appreciates, and saw this as a huge opportunity. So I thought this would be really neat to work on for a few years.
Well, I'm glad you work there. As the president of Galaxy, what do you want patients to know about Bartonellosis?
What do I want patients to know about Bartonellosis? I think more broadly, I'd like to focus on an awareness in the general public. And I think we have a very low awareness around the important role that the environment plays in our health. So we identify as a One Health company. We follow One Health principles. We do testing not only for human health, but also animal health. I just really want people to know that when you get a flea infestation, you should be worried and you should be trying to clear it up really quickly, and that fleas can transmit disease. And if things go sideways, right, that there are companies like Galaxy and labs like NC State out there who are experts in this area and can provide guidance on diagnostics and the potential disease involvement.
So you mentioned One Health, and Galaxy is a One Health company. So how does that guide Galaxy's business practices? And maybe you could explain One Health for our viewers.
One Health is a principle based, it really is an international movement among researchers looking at the intersection of human health, animal health, and health of the environment. But we have this kind of disconnect in the research world where, veterinarians study disease across different species. They understand pathology in a way that human health researchers don't. And comparative medicine, where we're sort of pairing animal and human health together, can provide some tremendous insights. And there have been some great books published in recent years that really illuminate that idea. Zubiquity is one that comes to mind right off the top of my head. And so that's what One Health is for me. And also when we're thinking about the environment and the impact as a society that we're having on the environment, land management, are we mowing the trees down? Are we building houses in the middle of forests? And all of these types of social patterns actually impact disease patterns. And this has everything to do with being sort of an emerging infectious disease in an area of medical discovery. We don't have all the answers. We know Bartonella, for example, is in the same ticks that transmit Lyme disease, but there's a whole area of research called vector biology that, that doctors, medical experts wait to see what the vector biologists have to say about whether the pathogens are actually transmitted from the vector into a host. Again, under One Health, we're bringing all the researchers involved in that problem together.
Instead of having them all be in their own
silos. And not necessarily focusing on or producing research that really solves real world problems. And that is a problem that plagues medicine, right? The physicians have one set of questions, patients have another set of questions. Meanwhile, the researchers are off trying to pursue, interesting, novel, advanced ideas that are currently being funded, because the funders have a huge influence over what research actually happens. Yeah. But it doesn't necessarily have anything to do with patients and physicians and the problems we need answers to immediately.
Patients, they're really suffering, and they often wonder why mainstream medicine has been so slow to recognize chronic manifestations of Bartonellosis. So how would you explain to them the main barriers to moving the needle and having it be recognized?
Where Bartonellosis is concerned, it's interesting. We have a lot of conversations with other researchers, with regulators, with physicians, even with other medical researchers, and there is a lot of interest. The pathogenesis research in general is super compelling, but there aren't clear funding sources for Bartonella research. And this is something we've been battling for 10 years, because as you know, what kind of sets Galaxy apart from a lot of other labs out there is it is about transforming clinical care, and that requires research and publication, and that is core to our strategy. We also think, by the way, that produces a better, more reliable, better understood lab offering as well. It's important that people understand how tests compare to other tests on the market, and that's something that we're trying to do through research.
Well, I have to say, I appreciate that Galaxy puts so much of their resources back into funding high quality research. One thing is that Bartonella is often considered a co-infection of Lyme, and one of the reasons I started my channel was because of what I consider that to be a misconception, because recent research shows that Bartonella can cause severe disease manifestations in individuals without Lyme. So what do you want patients and maybe even doctors to know about the difference between Bartonella and Lyme, or, borreliosis?
Right. Well, I'll say that historically in infectious disease, the way diagnoses and pathogens work is that there's usually one pathogen causes one disease. And the research coming out of vector-borne disease is really challenging that, particularly in tick-borne diseases, and certainly for borreliosis and for Bartonellosis. Right? Those are the technical terms for Borrelia infection and Bartonella infection. What does it say in the medical textbooks? In the medical textbooks, for Bartonella, it says Bartonella Hensley causes cat scratch disease, which is a collection of persistent lymphadenopathy, fever of unknown origin, just these non-specific types of immune responses. And Bartonella quintana causes trench fever, which is a slightly different collection of non-specific and symptoms, immune response. And then Bartonella bacilliformis, which is only really diagnosed in South America. We've only seen it in North America when people returned from, Peru, Ecuador. And I think that it's this one pathogen, one disease model is really problematic. Because what we're learning is that multiple species could be involved, and that for a doctor to narrow a diagnosis down and really fit that diagnostic criteria I'm tripping over my words now, fit that diagnostic criteria really tightly is actually the hardest part of a physician's job. You know, Bob Mosiani, who is our medical director at Galaxy Diagnostics, likes to remind people that there, you know, is a really short list of symptoms and a really long list of potential causes.
Mm-hmm.
And a doctor's job is to try to take, you know, those list of symptoms and rule stuff out until they can figure out what it really is. And this is something, so there's a lot of overlap in symptomology between borreliosis and bartonellosis. I'm going with the technical terms.
Yeah.
Bartonella and Borrelia, by the way, are known for asymptomatic infection, which means you could be infected, you can test positive for antibodies or potentially for the pathogen itself, but whether that pathogen is actually causing your symptoms or not is a clinical call. It's up to the doctor to determine whether this is, you know, this is a clinically actionable result. So I think that that's what I want physicians to know, what I want patients to know, is that there are multiple pathogens involved and may be involved in some of these diagnoses, and that the diagnostic categories are very loosely defined, and that we have to keep an open mind, and we've got a lot to learn.
That we do. So now let's learn about Galaxy's 2 different type of tests for Bartonella. So I've talked a fair bit about these on my channel, and so I'm going to link for our viewers in the top right corner of your screen and down below my videos on that. But Galaxy has their immunofluorescence assay, or IFA, and their triple draw enrichment polymerase chain reaction, or ePCR. So what would you like patients to know about these tests? And can you also explain the BAPGM to us, which stands for Bartonella alpha proteobacteria growth medium?
You know, we get lots of questions every day at Galaxy around infectious disease testing, and it isn't just patients who are overwhelmed with the complexity of testing. I think we're living that real time right now in COVID-19, right? That there are 2 basic approaches. You go indirect or you go direct, right? Indirect tends to be serology testing, or what we call antibody testing, where we're looking for evidence of an immune reaction against infection. You know, these indirect tests can be very specific to infection or non-specific, right? When you're actually testing with an antigen, with the bacteria itself, and looking for antibodies in a patient sample, we use whole antigens. You know, it comes with a risk of cross-reactivity because our bodies, you know, develop antibodies against surface proteins on pathogens, and those surface proteins can be shared. So those antibodies can get confused. So sometimes we can produce false positives. In Lyme disease testing, you may know it's Lyme Western blots, for example, are famously cross-reactive with Epstein-Barr virus. And that's a real problem because a lot of people have been infected with Epstein-Barr virus and have antibodies. So you know, the whole false positive, false negative. The other part of the problem with antibody testing is that it really only tells you that the patient's been exposed. It doesn't say that you're actively infected, and antibodies can persist in the body for a long time after the point of infection, right? But then there's lots of evidence that certainly the NC State team has produced showing, and numerous case reports have supported, and that's this idea that people with no or low titers can actually have DNA in their bloodstream. So how useful is antibody testing? And then you have direct detection methods. This is where the entire field of medicine is moving towards. We need direct detection methods. PCR is the most common direct detection method, and that can go after the DNA or RNA of the pathogen. And that says we found this genetic material in the patient sample. Now, whether it was alive or dead, we're not sure. We just know it was in the sample, it was detectable. Historically, the gold standard for diagnosing an infection is to be able to isolate and grow the pathogen on a culture plate, right? So you show that it was in the patient sample and that it was growing. And there are concerns around that, like contamination. Was the pathogen really in the patient sample? And we have microscopy too, which is another direct detection method where you can see the actual pathogen in a blood smear. The problem with PCR microscopy is that they often don't pick up like low yield infections. So infections that are pathogens that are in the patient sample at really low abundance, or that are intracellular, like Bartonella is intracellular. Borrelia is actually, if you see a picture, you have a cell and Borrelia is like huge squiggle next to the cell. Well, Bartonella is like, a tiny, tiny, tiny dot on the outside of a cell. Bartonella are not observable in a blood smear. So you see a lot of these labs who are doing these blood dot assays and low microvolume assays. You're not going to find Bartonella using that kind of an assay. It would be a 1 in a million chances. With Borrelia, you have a higher likelihood if it's in the blood. The problem is Borrelia doesn't stay in the blood, right? It tends to corkscrew its way into tissue, and it can come in and out of the blood, but not on a routine basis. So how do you design tests? The answer is, at Galaxy, we're all about what the pathogenesis is of the organism, how it behaves in the body, where it is, how do we design testing techniques in order to increase the odds of actually directly detecting, material of the organism in the patient sample that we're obtaining. And that can vary by sample type, but of course, like, everyone loves the idea of being able to use a urine test, except veterinarians, by the way, because it's a little harder to obtain urine from their patients. So everyone loves the idea of being able to do, a blood test or a urine assay. The problem is that depending on the biology of the organism and how it behaves in the body, you may or may not be able to find direct evidence. And so that's our problem set. That's what we're working on. We've made gains on Bartonella detection, direct detection, so PCR, by working with a sample enrichment method. And this is not unheard of in medicine. And that's all we're doing. The difference here is that Bartonella are extremely slow growing, and we have to incubate, so culture, enrich for a week at least. And that in research, we even do it longer. But it was a huge great gain, because before it was taking 6 to 8 weeks to plate culture the bacteria. And with BAPGM, it's a fantastic research tool. It increases the odds of being able to isolate bacteria for growth on plates. It increases the odds. It overcomes sort of a sampling error by increasing the bacterial load in the patient sample. So that was our big advance. We're not stopping there. We're working with other sample enrichment methods that will be less expensive and cheaper, and we're hoping to make some gains there. We have a new type of PCR that we've been working with for over a year, and the results are fantastic. We'll be launching a new and improved Bartonella ePCR by the end of the year. We're going after more targets than just DNA. We're also looking for, different proteins that may be good biomarkers that we may be able to also enrich or concentrate and increase sensitivity of detection. And so that's really what Galaxy is about.
So this new ePCR test, can you tell us how it's different or better? Or is it top secret?
It's basically digital PCR. It's used heavily in cancer. So again, in cancer, researchers are right up against the limits of detection, trying to find minute, like, gene expression and a sample full of a lot of other host genetic material. It's kind of like a needle in a haystack. And so digital PCR is a game changer that way. It's, instead of one reaction on each aliquot, we're running like 20,000.
Okay.
So much more. It's like taking a haystack and spreading it out over a field and going all archaeology on it, right? Looking in, like, making cubes and looking in each cube rather than attacking the entire stack to try to find that needle. We developed this method. We're working with a team at Duke on an endocarditis application, because that's our focus right now, is can we help people obtain earlier diagnosis of Bartonella endocarditis. Bartonella is the #1 cause of culture negative carditis in the United States. And and doctors would like better tools. They'd like to know whether it's an infection or not before they go in and replace a valve.
You kind of touched about testing with a microscope. And I have seen that some patients get testing, and I don't know from what type of people, but people who are claiming that they're visualizing the Bartonella, or sometimes I hear them being called Bartonella-like organisms in their blood. So how valid is this type of testing, and what kind of microscope would be needed or an assay to really do that type of testing?
Well, first of all, I'll say that everybody likes a picture, right? We like photo evidence.
Yeah.
But not all microscopy methods are the same. They're not equal. So for example, you go back to a very rudimentary blood smear. You've got a fairly low powered microscope compared to, confocal microscopy, for example, or some of the high powered technologies we have out there. It's a fairly low powered microscope, and that with staining, you can find Borrelia in samples that way. You can't find Bartonella in samples that way, or really tiny pathogens. So the challenge there is that just because you see a spirochete on a slide doesn't mean that it's a spirochete. So when you're dealing with these microscopy methods, the staining is really important. The fluorescence, increasingly, we're seeing, especially, I know Marna Erickson at the University of Minnesota has done some fantastic work combining these fluorescent tags that are specific to certain organisms with confocal microscopy. And it's so expensive to do, though, even on a research basis, it's like a, direct cost, just time and materials, about $1,000. But if you're going to go after microscopy anywhere, you really want to know, you probably want PCR confirmation that what you're seeing is actually what you're seeing. And if you can see it on a slide, you can PCR it. And I don't know any labs out there that are pairing microscopy, just like dark field or straight microscopy with straight imaging with PCR, but certainly for validation purposes, that's something they would need to confirm that they're actually seeing what they think they're seeing. And there are lots of different spirochetes in and around the human body, and there can be other types of tiny bacteria that look like Bartonella that cling to the outsides of cells, right? When it comes to the more sophisticated or advanced techniques that are in development now, like the fluorescent tags and confocal microscopy at University of Minnesota, these approaches are incredibly exciting. It's just that these technologies, it's very, very time consuming. It needs a high level of expertise. The materials are incredibly expensive. The equipment's incredibly expensive. And, it's going to take time for that kind of testing to be validated, to be affordable for patients. For now, fantastic research tool, which is why Ed Brachford, Ricardo Maggi, Galaxy co-founders of mine have been working with the University of Minnesota team on this particular technology. Dr. Moziani has formed a lab called T Labs, where they're starting to do some very early stage research using this technology. It's research use only. It's not a standardized assay. It hasn't been, tested inside, outside, and upside down. The idea that if we're not finding it in blood, but we could do a tissue biopsy, like a skin biopsy, image it, and and see what our fluorescent tags can pick up. That's really exciting.
So I think it's hard for people who don't have a real deep understanding of microbiology to understand like which type of microscope might be valid and what might not be. So would you say, like, unless you have like a confocal laser microscope, you really can't see Bartonella because it's too tiny?
Yeah, you're probably not seeing Bartonella, and it takes a particular staining method. The other concern, of course, is, around, lab quality, the engineering controls, the prep. People running microscopy in their garage or basement is a big concern because potential for contamination is quite high. That said, you do hear reports of people saying, I'll put my blood under a microscope and it's full of organisms. There's a lot of stuff there. Well, that is actually being confirmed by microbiome research. Microbiome researchers are shifting gears and starting to look at the possibility of not really a blood microbiome, because a microbiome is a healthy state, but rather the prevalence of, higher orders of microorganisms in blood in people who are sick. And so this is actually a couple of studies that Ed and Ricardo did using BAPGM, and it grows a lot of stuff, right? So they were enriching samples and then doing a blast analysis to look for DNA of different species. And in sick dogs and sick people, they were coming out with a long list. And these are 2 published studies, so they're out there for public consumption. And so there is, and this is consistent with the findings from like leaky gut. So a lot of what labs are finding, there's a lab up in Virginia doing some really interesting work. Apiriomics is using next-gen sequencing to look broadly for pathogens. Again, research use only, great research tool, but this is not a CLIA diagnostic. But they're really driving some discovery work, research, and showing I've heard reports they're finding that people who are sick often will have maybe what we can call a pathobiome, right? This is this concept that's emerging out of medicine. There's a French team who's done some publications on the pathobiome and tick-borne disease, that idea that you get a tick contact and all of a sudden you've got multiple co-infections or pathogens in your blood, and it's leading to this complex disease state. But it's also in more mainstream medicine, the pathobiome concept is there when we're talking about, the shift of the healthy gut microbiome into a pathobiome. It's kind of like, when things go bad. And then septicemia research is showing that people who are septic in the hospital, and they're trying to figure out where a lot of these pathogens are coming from. Most of them actually seem to be coming from the gut, which again goes back to this hypothesis, this leaky gut hypothesis that everyone was poo-pooing 10 and 20 years ago. Now we've actually got some good scientific evidence suggesting that when people get sick, the gut goes sideways, things leak into the bloodstream, and it makes you feel bad.
Mm-hmm. Well, judging by how I feel, I'd say my gut is pretty leaky.
Yeah. Again, this is all new emerging research, and most physicians are not reading medical journals. We are producing new research findings around health and medicine faster than we can translate them into clinical practice.
What are some of the studies that Galaxy is currently working on, and how do you think that these will further our understanding of Bartonella?
We're working on infective endocarditis, has the highest mortality of any disease presentation of Bartonella that we know of, right? Mm-hmm. So up to 20%, 1 in 5 people who are diagnosed with Bartonella endocarditis don't make it. We're also working on arthritis. We continue to work on the rheumatology and arthritis studies, looking for, evidence and joint fluid and studying different infection patterns, pathologies. And then we're also looking at neurological manifestations of Bartonellosis. Some of those are kind of the heavy seizure, epilepsy kind of presentations, and some of them are more neurologic function, neuropsychiatric types of manifestations. We've had couple of case studies published implicating Bartonella, for example, in schizophrenia patients, which was already known, by the way, that schizophrenia patients, that there was a high correlation with cat ownership. I've determined a long time ago that our crazy cat lady stereotype may have some scientific basis to it. And then the fourth stream of research is cancer. So Bartonella is a strong oncogenesis candidate because of what the pathogenesis research says it can do. And this is not to alarm anybody. We think it's probably a rare event, but it's kind of like HPV. It's, if it's there in a lot of people, most people, it's not going to be a problem. For some people, it could result in tumor growth. And so Bartonella can cause a long-lasting intravascular bacteremia, so intravascular infection. It can invade the endothelial cells and a broad range of other cell types. In the endothelial cells, it actually can trigger cellular proliferation, but it can invade a broad range of cell types. In other words, it has a way to get into cells. This is very unusual right, for microbes. And so these bacteria can infect the red blood cells, infect the endothelial cells, and the endothelial cells that line the vascular system. It can suppress cell death, so apoptosis, so it can prolong the life of the cell. And it can flip the switches, the DNA switches, to trigger cellular replication and growth. And there's research showing it can actually lead to the growth of new blood vessels. And so now the question is, it can cause tumor growth, but can it cause, what relationship does it have to malignancy? There's some research going on right now with a couple of different cancer models, 2 different types of cancer, where we're looking at those genetic triggers. Can it flip the switches that actually cause malignancy? So those 4 areas.
All very important areas and very interesting. So about transmission, it's a complicated topic, and I've done a couple of videos on that as well. I don't think that the research is completely definitive yet. And then I think that leaves room for both patients and doctors to speculate, and they have strong feelings on where they got it or how they got it. So some people believe that Bartonella can be sexually transmitted, and I haven't found any compelling evidence for that. So I was wondering what your take on it was.
So we don't have there's no evidence in cats or dogs. That's usually where the Galaxy team starts with. Do we have any evidence in animals? Has anyone looked? Have they found it has it been found in semen or vaginal fluid? And the answer is no. That said, it's been certainly detected in the saliva of cats, dogs, and people. But I think, people ask me this question all the time. The answer is we don't have evidence, right? What we do know is that Bartonella needs access to the bloodstream. So, for example, how do we get it from flea? We think about ticks biting us and injecting us with the bacteria, right? That's usually the way the vector biology works for ticks, but fleas and lice do it a completely different way. They take a blood meal, and just like a baby, when a baby feeds, a baby poops. And so these fleas and lice, they take their blood meal and then they poop, and then you itch, and then you're scratching it into your you're scratching it into your bloodstream. So yeah, we think that the odds are extremely low. We think the higher odds are that people share environment. So in the clusters, the family clusters, the workplace exposures, those studies have usually found that there is some kind of vector. There's often animals and people involved, and that supports the case that you're being exposed to something in your environment that is leading to the infection. And for the Galaxy team, the answer is going to be in the science. Have the studies been run yet? If there is evidence, how compelling is it? What does good evidence look like versus circumstantial evidence? But that's so we don't see any evidence for it yet. That doesn't mean that it is impossible. We just haven't seen any evidence.
And just because it's found in saliva doesn't mean it's viable necessarily.
Correct. It doesn't mean it could be in cat, and that's really important for cats and dogs, right? Where we first found detectable, and it's probably from how they clean. They're licking it off. They're licking flea poop off their fur.
Yep.
And that's where, and it's floating around in their mouth, and that's where we get it from. There's also, the FDA published a study a number of years ago showing that Bartonella is in dust mites. And so if it is in the dust mites, and we're ingesting that in different ways, what are the odds? Again, extremely unlikely.
Yeah.
So we think the odds are low.
Yeah. And you helped me a lot on my videos and understanding the science, and you framed it to me as probability versus possibility. So, obviously we can't say it's impossible at this point, but it appears that the probability is low for sexual transmission, as opposed to getting a flea bite and then scratching the poop into your wound.
Another thing I've learned is that if you have a sick animal in the household, you could end up with sick people. because those animals are sentinels. They spend more time in the outdoors around your house. They spend more time in the corners of your house, you know, and I just think that's something we should all be paying attention to.
Mm-hmm. Agreed. So about the cancer research that you were talking about with Bartonella, was that having to do with Neil Spector? And for our viewers that don't know, Dr. Neil Spector was a beloved figure in the Lyme disease community and a respected cancer researcher, and he recently passed away. And I know that you and he were good friends, so I'm sorry for your loss.
Yeah, Neil was a very, very, very special man. He was close to everyone on our team. He just was amazing. I did a blog post, one of the hardest things to write.
Yeah, I read it.
You know, an angel on earth, right? So personal, on a personal note, he was just amazing, wonderful family, and really was a game changer for us at Galaxy on a professional level as well, because he listened to our story and we really talked about what our concerns were, not just about borreliosis and Lyme disease, but also Bartonellosis and the potential links to chronic disease. And he was particularly captured with the pathogenesis research on Bartonella, right? Again, unlike Lyme, it's intracellular. Unlike Lyme, it can actually flip DNA switches in the cellular, at the cellular level, and it can cause tumor growth. And of course, Neil being Neil said, well, I know cancer. I think we need to figure out if that's what's happening here. His area of expertise is breast cancer. So he and his team at Duke worked with a team at NC State to set up the first inflammatory breast cancer model. Now, IBC, inflammatory breast cancer, are highly vascularized tumors. So it seemed like a logical place to start. Bartonella had already been detected in some IBC tumors, so we had some case reports there that were helpful. And so yeah, that's what we've been doing with Neil. Separately from that, NC State's also been running a cancer cell model for hemangiosarcoma, which is a very common form of cancer that affects dogs. Again, very vascular tumors, and that research has been supported by the AKC, the American Kennel Club. Neil's research was supported by the Cohen Foundation. He's also been working with some others at Duke on an immunotherapy approach to treating Bartonella and Borrelia infections. And that research continues to go on. And again, Ricardo was working, and Jen Miller have been working with that team to get them microbe smart. And we're very excited about the potential, right? That's that whole idea of harnessing the immune system to target the infection the same way that cancer therapies are using immunotherapy to target the cancer cells. So yeah, Neil was amazing. Can you imagine? I mean, just in a very short time, he wrote a book, he got his story out there, he organized 2 major research programs at his lab, organized the funding to support those. And I can't say enough how impossible it is what he did was to organize the funding to support the preliminary research on both of those. So he was a special person, and we're working our tails off to make sure that these research projects continue on. And there are a large number of people in the Lyme community who are working very hard to raise funding and ensure that his team can continue their work as well.
So this sort of relates to cancer in that it's a therapy that is supposedly being used for cancer, but also bacteria and viruses, but it's a bit of a controversial one. So it's called supportive oligonucleotide therapy, or SOT. So what is your take on the viability of SOT as a therapy for Bartonella, or at least in its form now?
Right, so there is science supporting this approach, to treating a number of different conditions. I'll say that the review articles I've read on it have suggested that the efficacy is really spotty for different for different applications. So I think it's scientifically credible, but I think that there's such variability across applications that a lot of research is needed to support the application for specific diseases. And I haven't seen any research, and I'm not aware of any clinical trials going on right now, to determine whether that approach is effective for treating Lyme disease or Bartonella infection more generally. And I'd like to see that. But then I feel the same way about, herbal therapies, ozone, hyperbaric. There are all sorts of, the stem cell therapies. There's all sorts of expensive therapies out there where claims are being made around how useful they can be for treating patients with these diagnoses, and I just haven't seen any science to support it. And I really hope, that they are partnering and working to bring some answers for patients on that.
Yeah, at least in vitro efficacy at the very least, which I haven't seen. Are you okay?
Yeah, sorry. Yeah, I know, and I don't have water here. I think you're spot on, Jake. I think that when you're working in an area of discovery like this, where the science is at an early stage, but the patients are finally have some hope, there just tends to be like this. It's a bit of a gold rush. And this is, very simply why the FDA regulates marketing.
Mm-hmm.
They don't usually take an interest until the number of people being affected is really wide. Take stem cell therapy for example. There are clinics, and you can go and get treated for $40,000 stem cell therapy, and it will cure you of Lyme disease. Will it? There are no case reports. There are no studies. Yes, there is some basis of science supporting the use of stem cell therapy for certain applications, but the outcomes are all over the place for different applications. And meanwhile, the marketing campaigns are going on.
I mean, I know what it's like to do an experimental therapy, and I'm doing it right now. Patients are desperate, and so.
And you know what, Jake, I think you should have the right to try something, under full consent, under full disclosure. I just don't know to what extent the information is available to help patients make that determination. And that's problematic. So my advice is for patients is buyer beware.
Yep. What do we know and don't know about Bartonella and autoimmunity?
What do we know? So autoimmunity is a diagnosis of exclusion. So any autoimmune diagnosis, there are very detailed criteria and test results and that are usually combined to determine whether a patient has autoimmunity. But for example, if you have a diagnosis of something like, autoimmune thyroiditis or autoimmune hepatitis, the answer is they looked for anything that could be causing the problem. They looked for infections, they looked for stuff, and they couldn't figure out what could be causing it. Nobody's looking for Bartonella. That's one of the problems, right? So I tend to talk about those types of diagnoses as immune mediated rather than autoimmune, because it's a curious question about what autoimmunity really is. The body attacking itself, and there's usually probably more going on under there than you think. And I think about, for example, how diagnoses like chronic fatigue and fibromyalgia have kind of evolved over time, and doctors have a good understanding now of the types of conditions that can contribute, the types of problems that can contribute to what looks like chronic fatigue, what looks like fibromyalgia. And so a large number of patients, if they have, for example, take fibromyalgia, a dysfunctional thyroid can cause the types of symptoms that fit the fibromyalgia diagnosis. And so you want to make sure that all of those tests, that deep dive actually happens. And of course, you might find a doctor who can do the deep dive, but do they know about some of the new findings that are coming out? Do they know to ask? This is one of talking to disease advocates out there. I was thinking about a conversation recently I had with Amy LaBeouf, who started the Louisiana One Health in Action group to
Also
focus on
moderator of our Facebook group.
yes, and moderator of the patient forum. One of her deepest desires at this point is to see physicians ask patients about vector exposure, animal contact, and travel history. It's like, if there's, what do you do for a living? Do you work with animals?
Yeah.
Do you work outdoors? Do you have, what do you do for fun? Are you a hunter? Are you a camper? Are you a hiker? Is there any chance that you could have been exposed to vectors? Did you have anything unusual? Because she thinks, when she tells her story about her son, Jack, she talks about the fact that she never thought that playing with kittens and a cat scratch could lead to what he went through. And, and the answer is yes. And it can be a long time before symptoms present. But if you don't know about it, you don't know about it.
Yeah, so I want to get some heavy hitting questions in before we end. So one is about striations. It's another not very well understood topic in Bartonella. And just for my viewers, I think there's a lot of confusion over the word striation. So that's the medical term, as I understand it, for stretch mark, just like fasciculation is the medical term for muscle twitching. So what do we know about these marks and what do we not know?
Yeah. Mostly we don't know a lot. They've just been observed in patients who've tested positive for Bartonella bacteremia. I think the group leading a lot of this research, so again, Marna Erickson out of University of Minnesota, Ed Breitschwartz, we've done the NC State team, Ricardo have done the PCR verification on some of the tissue and blood samples from those studies. The answer is doctors are seeing these marks a lot in the patients who are testing positive and are sick. Mm-hmm. And so they're wondering, what's the relationship between the symptoms, right, the marks, the cutaneous presentation, the skin manifestation, and the positive test results. And so what Dr. Erickson and colleagues are doing is that using the imaging is they're actually, she's been working in the Department of Dermatology at University of Minnesota School of Medicine for a long time. So her expertise is in imaging skin and understanding how, what cause and effect around different types of skin presentations. So she's trying to figure out what these bacteria could be doing to the collagen. And so that's kind of what we know so far, is that they're there and they seem to be doing something, but exactly what that disease mechanism is undefined. We also don't know, how common is it? We see questions all of the time on the patient forums where people say, can you have a Bartonella infection without these? And the answer is, yeah, the same way you can have a Bartonella infection without Lyme disease. I think it's really important to understand that this is a complex story they're unpacking. Meanwhile, there are a lot of physicians out there who see that as an indication to look for Bartonella in a patient, and we just don't know. We just don't know if it is a good marker or not right now. Mm-hmm. So physicians do what they need to do in order to deliver good clinical care, and as researchers, teams are digging into that to see if they can unpack that and identify if there is actually a correlation and how prevalent it might be.
And so correct me if I'm wrong, but I believe I've read that Bartonella can sit outside of the vascularization in the collagen. So it's been visualized there. So perhaps it's damaging the collagen in some sort of way, and striations develop.
And it's also, and you can also see it actually in the vasculature. I mean, it's that you see it there more, but then you can see it dispersed out into
the collagen,
the tissue and the collagen. It's really challenging. The problem there is, again, you see these striations on a lot of people who don't have symptoms, aren't sick. Mm-hmm. But again, if asymptomatic Bartonella infection is common, then, I mean, there's a whole story to be unpacked there. None of this necessarily says that it isn't, that these tracks aren't don't involve Bartonella. One of the big questions around, we know that we find Bartonella in diseased joints, right? And so there's a bit of a chicken or egg problem here. It's kind of, and I think of the plant, right? Is it when the plant gets sick, did it get sick because the white flies were on it, or did it attract the white flies after it got sick?
Mm-hmm.
So we're looking at that same kind of puzzle. Are we finding Bartonella in tumors and diseased joints because it's drawn to these vulnerable areas, or did it cause the disease process going on in these areas? And we just don't have an answer to that right now.
Well, hopefully we will one day.
No, but we're getting into that whole question of host response, right?
Yeah. Yeah, and I think that's what's really confusing to patients. Like, how can one person like me have no brain fog, no fatigue, but my mast cell activation is quite severe and refractory, and I have a lot of bodily pain. And then another patient has striae and schizophrenia and diarrhea. So I think that's very confusing to patients, but it's the interplay between the host and maybe the pathobiome.
Right, exactly, and it can be a perfect storm of factors. The host response factors are really important in the world of infectious disease. It's rarely the pathogen that is causing the disease. It's usually the immune response against the pathogen that's causing the disease, right? That goes back to that autoimmunity hypothesis. Mm-hmm. If you can't figure out what the immune system's attacking, then it must be attacking its own tissue. And so, the host response part of a systemic infection like Bartonellosis can result in, or like, Lyme disease can happen. I think that when we're looking at a systemic infection, you're looking at involvement in multiple body systems. You're looking at different presentations because the individual may have different susceptibilities, either based on genetics or based on underlying conditions or medical history or lifestyle. Like you eat a lot of crappy food, so maybe those are the people who get the GI problem.
Guilty.
But it begs the question, right? And we don't know any of that because there's no funding to do that kind of clinical research. And like at Galaxy, our whole thing is if we can come out with better testing so we can better define the patient population to test, then we're going to be in a better place to help make those studies happen. And, and we're excited about some of the novel therapies coming out because some of those groups are coming to us and saying, will you work with us to help do the in vitro research? Will you help us figure out the animal models? Will you help us do the clinical trials?
Yeah.
We need diagnostics before we can answer a lot of questions.
Yep, it's kind of the starting point for everything. I guess the last question I have for you is, I always want to leave my viewers with a sense of hope. So what are you hopeful about in the Bartonella field as it relates to patients?
I have to say the one thing I'm most hopeful about is I'm most hopeful about patients organizing. We are seeing the beginning of fundraising, specifically, for example, for North Carolina State Medical Foundation to support Ed's lab, or for University of Minnesota supporting Marna's lab up there. Mm-hmm. I'm really excited about that because I think that it's really important for everyone, as much as we don't want to accept the moniker of rare disease, right? Bartonellosis is a rare disease. Mm-hmm. And if we start identifying ourselves as a community, as members of a rare disease community, whether we're the patient, the advocate, the family support member, the physician, the funder, I think the answer is that's where the future lies. We've got to organize around this problem. We've got to raise the funding. We've got to identify, the labs who are really driving discovery around this, get funding into those labs to move the research forward. From Galaxy's perspective, we're getting funding now that helps us generate preliminary data that makes us eligible for federal funding. Yay. And we've been going after federal funding for a decade, right? And a lot of what we get back from the reviewers is, this is really compelling. We look forward to seeing your next application with more data. And then we're sitting here going, where are we getting the money to pay the people and the materials and the equipment and facilities to get this data generated? Mm-hmm. I'm excited to see the patient communities organize. I'm excited to see people organize around fundraising. you know, and brilliant moms like Amy, who not only, you know, has pushed treatment and recovery solutions for her son, getting the right diagnosis and getting the right treatment, but she's gone and started a nonprofit, and she's holding fundraising events, and she's out there talking to the people at the medical school and the veterinary clinics and the vet med school and in public health, and she's getting them all to show up in one place to listen to a presentation. And it's just, you're doing these videos and just one little bit at a time in the memes, right? Just getting that research out there. That's what Galaxy's been trying to do right from the beginning with our social media. It's a big, complicated story. We don't have all the answers, but we can get one study out there at a time. We can hold one event out at the time. We can support one study at a time.
Thank you so much for talking with me. I really appreciate it.
It was a pleasure.
And
excited about your move into interviews.
Me too.
We have to see who else we can talk into.
And I don't know if the camera picked it up, but in the beginning, my eyes started watering because I was nervous. But then I eased into it and my eyes stopped watering. So
nothing to be nervous about. Yeah.
But thank you so much. You know, we're just going to continue pushing this forward and getting somewhere for patients.
Fantastic.
Okay, well, I'm gonna hit stop recording now. Bye. Okay.
It says record.
Cool.
I didn't sign a release.
Is this how it's gonna be the entire time?
I'll be good.
What's the difference between the IFA and the ePCR?
And
Why didn't you turn off your phone when you were coming to meet me, Amanda?
and think about it.
So can you tell me the current accuracy rate of the triple draw?
100%.
How is Galaxy improving their tests? Are you working on new tests for Bartonella? If yes, how will it be available and different than the ePCR already in use?
Truthfully, I have no clue.
I can tell you are an invaluable member of Galaxy.
I am. I really am.
What are some studies that Galaxy is currently working on, and how will these studies further our understanding of Bartonella? Great. And some people believe Bartonella can be sexually transmitted. So what is your take on this topic?
You know what? You can get Bartonella in any freaking orifice. If you can stick something in it, you can get it. You can get it in your ear. You can get it in your eye. You can get it in your nose and your mouth. You can get it in your back and your butt kiss and your little pee hole. Oh, lips. There are holes there too, and it can crawl right back in.
You are getting worse. You are getting more terrible by the second. Okay, yeah, I totally agree. How did you come up with the name Galaxy?
Because it's out of this world, baby.